Well, we'll go ahead and get started if it's okay with everybody. You okay to go here, Al?
Yeah, go ahead.
Okay. Well, today this is being put on and actually was suggested by the Musella group and Al Musella, and it was a discussion with Matt Dunn and effectively talking about different types of combination treatments as we move forward in the DIPG challenge. For many of you, you may already be familiar with Dr. Matt Dunn. His official biography, and I'm going to read a little bit here, but he's associate professor and received his PhD from the University of Newcastle in 2012, and since has been decorated by over 25 national and international awards, including the 2019 NSW. That's was it New South Wales? Is that correct?
Yeah, that's the state we live in.
Okay, premier awards for outstanding cancer research fellow and a Young Tall Poppy Award in 2020. Professor Dunn has secured continuous fellowship funding from the Cancer Institute of New South Wales, the NHMRC Investigator Grant, a defeat DIPG.
That's our NIH.
NIH, okay.
That's our NIH, yeah.
And has also received funding since 2012. Professor Dunn leads the Cancer Signaling Research Group at the University of Newcastle and the Hunter Medical Research Institute, which focuses on pediatric leukemia and brain cancer research. His group employs sophisticated pharmacophosphoproteogenomics. Did I get that one?
Yeah, you got it.
Okay, to characterize the cellular signaling pathways dysregulated by the genetic individualities of a patient's cancer. This profiling strategy attempts to identify novel treatments, targets, and drug combinations to improve survival. Now, most importantly, though, he's also a father. Obviously, you fought the battle with your daughter, Josephine, and I believe she passed at 4. Is that correct?
Yeah. 3 months before her 5th birthday, yeah.
Yeah, so obviously Matt understands this battle. He understands it both from a doctor's perspective, but also from a parent's perspective. And certainly we all understand that as well, most of you that are attending here. What he's going to talk to us is about the future. What many experts out there believe is that this is going to take on a very similar treatment strategy to what we're doing with other cancers and try to bring other strategies together rather than really one singular cure. And so with that, I'm going to go ahead and turn it over to Dr. Matt Dunn, and we'll go from here. Thank you.
Thank you, Keith, and thank you, Al, for the invitation. And happy New Year's Eve for those of you who have reached New Year's Eve, and then New Year's Eve for those of you in the States. Yeah, so this is, I suppose, the culmination of 4 long years of hard work since Josephine was diagnosed with DIPG at the age of 2 3/4. And so, if I may start, I must mention my disclaimers that, of course, I'm a parent of a child lost to DIPG. And with the help of some wonderful family and friends, we established a charity at diagnosis, really, to help fund some of the research that Josephine was then able to benefit from, although transiently. And so this is Jojo, 6 months prior to diagnosis, scooting around on a scooter on the netball courts. And netball's a funny game that it's like basketball, but you can't run. It doesn't make any sense to me either. And this is a really important video. That's dorky dad on his skateboard, following her helicopter parenting, as always. And back here in the back right-hand corner of this video, you can see a large tree, and that's now where Josephine's buried. So this part of the world is very important to us. So many of you would know about DIPG, but for some of those that are tuning in to learn a little bit more, may I help? So diffuse intrinsic pontine glioma, which is also known as diffuse midline glioma, is a mostly pediatric high-grade glioma, so a glioma that's of a very advanced stage, and it's diagnosed most usually in the brainstem. And median overall survival, so that's the amount of time that kids survive after diagnosis, is a pretty robust 9 to 11 months. and we very rarely see any long-term survivors. And it affects kids at about 6 and 7 years of age. And I've noticed anecdotally, and I'm sure there's data to back this up, that kids that are diagnosed around 6 and 7 tend to do the worst. And the reason why the disease is such a horrific and harrowing experience for the child is because where it's diagnosed in the pontine region of the brainstem is a critical little organ that regulates life-sustaining functions, heartbeat, respiratory control. And then the motor neurons or the nerves that run either side of it are then squashed by the growing tumor, which then causes them to not be able to transmit the signals from the brain. And so you start to see a loss of motor function and sensory. And soon enough, the growth of the tumor interrupts life-sustaining functions. And the only current treatment for DIPG is radiotherapy, and that only extends survival by around 3 months. So we here in Newcastle and colleagues around the world are working together to develop treatment strategies to extend survival of kids with DIPG. Now, if we have to talk about the genomics or the genetics or the genes that cause DIPG, and it's kind of remarkable how homogeneous, which means there's a gene mutation that occurs in almost every child with DIPG, and that's a gene mutation, so a change in the DNA sequence to a gene called histone H3. And histone H3 is a critical gene for wrapping up all of our genes. So it's a fundamental unit of what we call chromatin, and this acts like a reservoir of DNA protecting protein. So it wraps up the DNA to make sure that the DNA can't be damaged, and so that every time a cell divides, the DNA is showing very high integrity, and then you don't get things like cancer. But when this gene is actually mutated, then the DNA becomes more susceptible to increased damage. And it happens in 2 of these histone variants, and in the younger children, it's usually in this histone H3.1, as we know, K27M, and then in the kids that are older than 6 and 7 and young adults as well, we see a mutation histone H3.3. And you can see that the survival for these 2 variants is somewhat similar, and we think that patients diagnosed with H3.3 mutations succumb at about around 9 months, and patients with H3.3 mutations succumb at about 15 months. And this is somewhat similar to DIPG or DMG patients who have no mutation in histone H3, but they have increased abundance of this gene called EZIP. And EZIP kind of acts within the cell in the same way that the mutation does. It kind of turns off some of the machinery that's required for the genes that drive cancer to be produced. And we don't know how that happens yet. And it's only a new classification system, but the survival here is about 15 months. But for the longer term survivors that we have for patients with DIPG or DMG, they are patients that usually harbor wild type H3, but that only happens in about 10% of cases. And what happens in a summary is that, as I mentioned before, this hose reel looking like structure is the nucleosome, and where histone H3 is housed, and the DNA is wrapped around. I'll just get the pointer out. And the DNA, as you can see, which is this grey cord, wraps around it, and so that these are the genes that give us our characteristics. So the genes that make us look like we are, the genes that are involved in metabolism, so breaking down of food and turning it into energy, and the division of the cells. And so usually, particularly in the developing brainstem, these proliferative genes, so genes that make the cells grow really quickly, are wrapped up nice and tightly. And then for them to be unraveled and then turned into a protein, a complex called the PRC2 complex comes and takes off these little balls, which are methylation marks, and the DNA can become unwound. But when the mutation happens, the DNA is always unwound, and these proliferative genes, things that drive cancer, are always switched on. We did a bit of work throughout the year, and this is a fantastic study by one of my PhD students in the lab, Isaac Finlay, summarized the recurring gene changes that make up DIPG, regardless of what type of histone mutation they have. And then you can see here, within this circle in the middle, these are the mutations that are seen across histone mutation subtypes. So 3.1, 3.3, and our wild type, but EZIP. And you can see here we have TP53, which is a tumor suppressor gene, PPM1D is a tumor suppressor gene, P10 is a tumor suppressor gene. And then we have 3 growth and proliferation genes, PIK3CA, EGFR, and FGFR1. So as you can see here, two things usually occur in concert with the histone mutation, and would occur after the acquisition of this initial driver mutation, which probably happens soon after conception. And that's the switching off of tumor suppression and the turning on of proliferation. And for interest, Josie had PIK3CA mutation, P53 mutation, a P10 mutation, and a H3.1 mutation, and a B core mutation, and a number of other things. And so this nice summary of the gene mutations that drive DIPG is summarized here in this nice journal article, and I encourage you to download and have a read. And this also gives us some of the perspectives of how we think the incorporation of multiple types of biomedical research techniques are necessary to really unravel how we treat patients going forward. But not only are there a huge burden of really aggressive gene mutations that give rise to DIPG, mutations that alone would cause other types of cancers and have very poor outcomes. And here we have DIPG patients that harbor, on average, between 4 and 6 of those driver mutations at diagnosis. But not only do we have all these different gene mutations, they combine in different way in different cells within the one tumor to drive different diseases within the same disease. I mean, that might sound a bit crazy, but I think of it like a suburb of a city. So we're all, for example, here in Newcastle, we're all Novocastrians, but I live in Dudley, and the general manager of the charity, Troy, he lives in Garden Suburb. We're similar, but we make up a colony of unique people that live in Dudley and Garden Suburb, and we all come together at some times, but we're all driving in different directions. And that's what happens with DIPG. DIPG is a really aggressive disease that's made up of a number of different colonies or suburbs or parts of the same disease. So targeting it with individual therapies, so precision-based therapies targeting 1 or 2 of those mutations, is unfortunately unlikely to work, because within that same disease or within that same tumor, there's multiple different colonies made up of different gene mutations that drive different rates of growth, that drive the ability for the tumor to avoid immune system detection, that drive the ability for the tumor to respond to different concentrations of oxygen within the brain stem, or indeed things that make the cells grow, such as insulin and glucose and fats. So it's a really aggressive disease. And although it doesn't harbor, the same number of mutations that you would see in a lung cancer patients, there's more clones. So the more different suburbs within that tumor that harbor driver mutations, so non-silent mutations in DIPG, than any other tumor type. So we call that a very high level of intratumoral heterogeneity. And not only do we face a huge number of driver mutations at diagnosis, the highest level of intratumoral heterogeneity of any cancer, but the contribution from the normal structures within the brain play a huge role in the aggressive nature of the tumors. And Michelle Mongi, who's obviously the absolute matriarch of DIPG research, has beautifully shown this in a number of ways recently, where it's not only that the tumor cells talk to each other, but the tumor cells talk to the normal neurons, and the neurons pass on signals and encourage them to grow. And then from that, we get the vasculature, which provides the tumor with nourishment. And then we add corticosteroids, which again, probably drive the growth of the tumor. And then we have radiotherapy, which also has an effect on the tumor, particularly the tumor is growing in low oxygen, or what we call hypoxic niche. And we've got some emerging data that we're working with a fantastic scientist in Israel, Oded, who's shown that the microglial cells, so that the brain's immune system, also drive the growth of the tumor. And that's some exciting stuff we hope to show you probably next year or so. So given all of these factors, the clonality, the number of somatic driver mutations, the different combinations of effects from the normal structures, genomically based monotherapies or even combination therapies are unlikely to improve outcomes. And so, we're kind of focused on research that we know we can get an effect from the DIPG cells. So what I mean by that is that we're using drugs that we know show good accessibility in the brain, and particularly the brainstem, and then have some effect, hopefully positive, in terms of the cells not liking the drugs, and then on response to those drugs, identifying what they are, and then coming up with strategies to combat the way that they become resistant to those therapies. So we're kind of thinking of it in a way where we almost want to shepherd the tumor cells to grow in a particular way. Hopefully some of them die from that single drug, but we know from what I just mentioned that one drug's not going to work. And then come along and target the DIPG's response to that drug, and hopefully kill off more of the cells. And then again, it might take another effect. So, using CNS active drugs that initially focus on energy homeostasis, so the unique way that DIPG cells produce energy to drive their really rapid growth, and then used in combination with the response, drugs targeting those factors. And then hopefully we've got a debulked tumor that we can then use a more sophisticated immuno-oncology approach, or CAR T therapy with Michelle or Nick Bentenza in Seattle, and somehow as a maintenance therapy, keep that tumor at bay. And hopefully in time, by hitting it in all of these different ways, the tumor dies out, and we start to see some long-term survivors. So many of the audience here would know all about ONC201. When Josie was diagnosed, there was only anecdotal evidence for its efficacy, and that wasn't even in DIPG yet. That was in a glioblastoma patient, this young lady who had almost complete objective response to the treatment for almost 3 years, and she received that drug once a week. And when they did some tumor sequencing, they identified that this patient indeed had a H3K27M mutation, and then hence clinical trials started to test ONC201 around America for H3K27M DIPG. And this is old data now, and I've seen some new data, but this is data from Carl Koshman's group in Michigan, who initially had assessed 23 brainstem patients and 27 thalamic H3K27M patients, and the response was 17.1 months and 28 months respectively. But I've seen new data recently with an expanded cohort, and I think there's 60 patients from each, and now it looks like ONC201 as a monotherapy is somewhere around 20 months for brainstem patients and not reached for thalamic, which is really showing that we're on the right track. We've got a drug that does something. Now it's up to us as a community to figure out how we make that drug work even better. Many of the audience here, including myself, would have heard about access to ONC201 through a German oncologist, and probably about mid 2018, 4 or 5 months after Josie was diagnosed, a beautiful mother from Portugal, Christina, had contacted me and asked me whether I could figure out what was in the capsules. And of course, being a biomedical scientist and having strong skills in analytical chemistry, I said, of course. And we started to assess what was in it. And we found that the German version of ONC201 was almost identical to the drug used in clinical trials. However, it was slightly less protonated, meaning that when you dilute it in water, it was a base, so it was insoluble in water, whereas the drug used on clinical trials is completely soluble. And and that the drug was encapsulated with a particular type of sugar called mannitol. And we still don't know why it was, but mannitol's been used in other studies to try and get drugs into the brain more effectively. So potentially it was done to increase its brain penetration. But clinical trials had already assessed the excellent brain penetration pharmacokinetics of ONC201 in glioblastoma studies prior to resection, where you can do a surgery on the tumor and show that, you know, with 625 milligrams of the drug, you could get almost 1 micromolar to 9 micromolar of the drug, which is more than enough to see a therapeutic effect in DIPG cells anyway. We collected the clinical data on 28 of those patients and families that had elected to purchase German ONC201, and analysis compared to historical controls revealed that the patients survived for a median overall time of 18 months. But a caveat that each one of these families and patients had combined ONC201 with all ranges of other drugs and therapies. Probably most commonly CBD was the most used drug in combination with ONC201. So the survival benefit can't be attributed to ONC201 as a monotherapy here. But I suppose even for the patients that succumbed within 9 to 11 months, as shown by this blue line here, you know, I know as a parent, you know, you would have tried all manner of different targeted therapies, combination chemotherapies, all sorts of kind of drugs to help your children, and yet the survival remains 9 to 11 months. One of the interesting observations within the same study that we also saw was that parents and families that elected to have reirradiation whilst on ONC201, the German ONC201, survived for 22 months, compared to those patients and families where no reirradiation was offered to their children, and that remained at 12 months. And this has recently just been published. And a sincere thanks to the team that worked on this paper and the clinicians that provided the patients they knew and their families that they knew had no other option but to purchase German ONC201 for the treatment of their children. But one of the things that got to us when we first started to use ONC201, and this is the ONC201 from Chimerix or Oncoceutics, is that there's a huge level of response to the drug. You know, this is a huge amount of drug here, and we're seeing most of the cell lines not reaching IC50, which is the half of the cells alive at that time. Some of them are super sensitive, and then there's some that are completely refractory. And this is again further reflected here when we measure, this is growth and proliferation, and when we measure cell death, and we can see that in a number of these cells, we see a slight increase in cell death, and others are completely ablated, which is great. But there is an inherent resistance mechanism that underpins resistance to ONC201. We tried to ascertain what that might be using a genomics-based approach, and we have some clues about what that might entail. We're still working on complete resolution of what we think at the genomic level drives resistance, and I won't talk about that today because there's some sophisticated studies going on, but that should be finished at the end of Jan. But we did use the unique profile of sensitivity of our cell lines to assess biochemically, so to figure out how the cells responded to the drug, try and give us an idea of what we should combine with it. At the time, and it still hasn't really been published, but we did make some suggestions within our latest German ONC201 paper that what the mechanism of action is within DIPG in terms of what is the drug target in the tumor in the brainstem. And so ONC201 is a dopamine receptor D2 antagonist and a mitochondrial protease, CLIP-P agonist. So that means an inhibitor of DID2 and an activator of CLIP-P. And indeed, that's what we see at a biochemical level, at the protein level. So the more DID2, so the more the dopamine receptor D2, the more sensitive the cells. And this is a bit confusing, but the more of this protein here, which is one of the proteins that make up the mitochondria that produces energy, the more of this, the less sensitive they are. And I'll show you what that means. So this is nice modeling work where we take both German ONC, so the protonated and the deprotonated versions, normal onc and German onc, and we bind them into the pockets of the proteins that they're meant to be targets of. So ClpP binds really nicely, DRD2 also binds, and the strength of the binding is about similar. But then we measure the effects by looking at what the effect is downstream of proteins we know are regulated by those pathways. So ClpP, so when you activate ClpP, you get degradation of this mitochondrial protein. So this ClpP is a protein that eats up other proteins. It's called a protease. And one of the major targets of it is this protein called SDHA. And you can see here that these little black dots mean that the more of the black, the more of the protein there is. So when it decreases like this, something's happening. You're getting less of it. So you can see that the mitochondrial electron transport chain, which produces energy, is getting eaten away by treatment with ONC201. And similarly, ERK signaling, which is one of these proliferative signaling pathways that's downstream of the dopamine receptor 2. So when this is activated by dopamine, you get ERK signaling. And when you treat with ONC201, you turn off dopamine signaling quite strikingly. So in our view, it's not one or the other, it's a combination of both. You're getting loss of mitochondria and you're getting loss of dopamine signaling. And it doesn't matter whether the cells are sensitive, as you can see here, or resistant. The same thing happens. And this is lovely work from Jason Kane at the Hudson in Melbourne, who's a great collaborator. And so he genetically deleted dopamine receptor D2 from DIPG 36 cells, which are somewhat sensitive, are quite sensitive to ONC201. And when he did that, the DIPG cells just died. So the DIPG cells need dopamine signaling. Whereas when he deleted this mitochondrial protease, ClpP, the cells, and then treated with ONC201, nothing happened. So it showed that ONC201 indeed does drive the activity of ClpP. And so ClpP is an important protein in this story, and we don't neglect DRD2, but we're still working on it. But what we've been able to show is that looking at sequencing data from about 400 patients, these are patients with different forms of central nervous system tumors. When we measure how much CLPP or ClpP there is in their tumors compared to your control and other types of cancers, you can see that in DIPGs and high-grade gliomas, ClpP is really highly expressed. There's a heap of it. So it kind of makes sense why ONC201 works more specifically in DIPG. And I wonder about the high-grade glioma. But you know, obviously there's been some clinical trials done in adults in high-grade glioma, and it hasn't been that positive. I wonder, and these are just pediatric patients, so I wonder if I compared the expression of ClpP in adult high-grade gliomas, such as glioblastomas, compared to pediatric, whether there would be a difference in ClpP, and that's something that we should do. But regardless of whether they're sensitive or not, ONC201 causes the production of free radicals. So how that works is that ONC201 enters the cell, it activates ClpP, as I suggested. That leads to the eating of the electron transport chain or mitochondrial proteins, and as a consequence, you get this production of reactive oxygen species or free radicals. And these are oxidative molecules that do a lot of things within the cell. And what became obvious to me when we look at this is that the cells here, which are our most resistant, are completely refractory to ONC201. They already have a huge amount of oxidative or free radical production, even in untreated circumstances. And we can drive that a little further with treatment to a certain point, and then the cells don't produce anymore. Whereas in the more sensitive cells, they produce less free radicals or reactive oxygen species, and then the drug makes them produce more and more. And then that probably leads to oxidative DNA damage, and the cells die, whereas these cells are resistant. And this is beautiful imaging from Javad Nazarian's group in Zurich and at Children's National that indeed shows you the red, which is the oxygen, the free radicals. So once you treat them, they produce heaps of free radicals. So to assess how we develop better combination strategies by treating on the DIPG cells with ONC201, we did profiling, also protein expression profiling of DIPG cells that were completely resistant to ONC201, but cells grown in different oxygen tensions. So those of you who know a bit about cell biology would know that the mitochondria uses oxygen to produce energy, and that within a tumor, there's a huge variation in the oxygen tensions of the tumor, depending on where the tumor is growing. And one suggestion is that the tumor grows faster than the rate of vascularization, so the rate at which the tumor can develop blood vessels to feed it air and oxygen and blood. And so they inherently become hypoxic tumors. And I've seen some data from Carl Koshman recently that also suggested that the patients that have a more hypoxic tumor do less well on ONC201. So we use these properties to try and determine how we best develop combination treatment strategies. And when we assess how the cells respond to 24-hour treatment with ONC201, we see, as before, most prominently the loss of mitochondrial function. So the loss of mitochondrial activity, oxidative phosphorylation, which is the way that the mitochondria produces energy. And then we had some interesting hits here with hippo signaling and amyloid processing, stuff that we're working on and might become important later on in my talk. But then the cells were able to upregulate protein synthesis. So they were able to, even though they weren't dying, they were growing, they produced heaps of new proteins to keep them healthy and happy and to grow even in the presence of the drug. And as mentioned before, we get this huge loss of mitochondrial proteins, and then we get increased activity of these transcription factors, which are responsible for producing new proteins. But when we take all of the data and we plug it in and we try and figure out what the central node of resistance is, this protein here comes to the fore, and this is called AKT. And this protein is the downstream effector of a PI3 kinase signaling pathway. And remembering back to Josie, she had a mutation in PI3 kinase, which regulated this protein, which meant that this protein was always switched on. So in recap, so cells that are resistant to ONC201 seem to have increased activity of PI3 kinase AKT following treatment. So if we look at that visually, if we take a snapshot here, these are resistant cells. We treat over a time course, we lose this mitochondrial protein. This IDH3B, which is also quite a very important mitochondrial protein, and we get increased abundance of this AKT protein. And these are all downstream effectors of AKT, and these are involved in the production of new proteins, the regulation of amino acid synthesis, and energy production and growth and proliferation. And this pathway also regulates the response of any cell to oxidative stress. So as I mentioned before, when we treat DIPG cells with ONC201, we cause the mitochondria to start to break down and produce free radicals. Usually in the DIPG cells, the cells would die, but in cells that are resistant, they have this mechanism of antioxidant response. And that's to produce this reductase NQ01. And I won't focus too much on this now, but this will become important when we think about how we combine multimodal treatment strategies going forward. And so a graphic of this, again, in summary, is that ONC causes mitochondrial dysfunction, which produces reactive oxygen species. This is then driving the activity of PI3 kinase, AKT, and growth and proliferation, and at the same time, regulating antioxidant response. And parents often ask me whether they should take vitamins and antioxidants and all these supplements. And we try to explore Boswellia and all this kind of stuff as well with ONC. My suggestion would be not to. My suggestion would be, if you want ONC201 to work, it needs to drive as much oxidative stress as possible without being rescued by antioxidants. That would be my take-home message. And we've done work with antioxidants to show that we can deaden the effect of ONC201. But given the increased activity of this PI3 kinase AKT signaling pathway, well, this then uncovered a potential combination treatment strategy. And at diagnosis, when we got Josie's biopsy sequence through the Zero Childhood Cancer Program, which is this fantastic translational sequencing platform where it takes kids with cancers and sequences their biopsies to determine the gene mutations that give rise to that tumor. We saw in Josie's tumor that she had heaps of events within her tumor that drove the activity of this PI3 kinase signaling pathway. And so I went on the hunt for a drug, and a lovely colleague had seen this drug GDC 0084 at a conference, and I looked into it, and It showed excellent brain penetration, pretty manageable side effects in adults with glioblastoma. And so we got the drug Paxalosib in the lab and we tested it, and we were the first to test Paxalosib in DIPG. And this is work for Ryan Duchatel, who's done a beautiful work on characterizing Paxalosib and is about to submit his findings for peer review. And we can see that in DIPG cells, they're much more sensitive to Paxalosib than the patients that were receiving the drug, the GBM patients. And it had a very potent effect on decreasing the abundance of this AKT protein and its downstream effectors. So to us, it made sense to treat these cells with Paxalosib in combination. And thanks to Chris Tinkle's work at St Jude's, we'd had a maximum tolerated dose. So Josie became the first kid worldwide to receive Paxalosib. This is work from Evie Jackson, who's used it in combination with ONCT-1, and this is combination index analysis. So what this graph means is that these are the different doses of ONCT-1 in combination with Paxalosib, which is down the bottom here. And so anything that's below this line of 1, and these are all of our resistant ONCT-1 lines, anything below the line of 1 is a synergistic effect. So when we combine the two, we get a great synergy even at low dose. And this is visualized here when we do colony forming assay, which is the same dose as used here at this bottom end. So the most physiological dose, so the doses we can get into children safely. And you can see the number of colonies that decrease when you use the drug in combination, whereas using the drugs alone, you hardly see an effect. So that's promising. But again, it highlights that there's clones or colonies or DIPG cells that are surviving even in combination. So we need to do more. And this is lovely work from our group in combination with the help of Shri Divi, who's at Children's National under Javed Nazarian. We tested the combination in DIPG6, which is our most resistant ONCT-1 line, and showed that the drug was quite effective at decreasing tumor burden, particularly Paxalosib and the combo, whereas there wasn't as much an effect with ONCT-1 alone, and that we could increase the survival of these mice using the combination quite dramatically, whereas the single doses increased survival alone. And the interesting thing, I suppose, seeing here is that although ONCT-1 didn't have much of an effect on these cells in vitro, so in the lab, when you gave it to an animal where global dopamine signaling would be happening within the brainstem, we now start to see an effect where we get an increased survival from the monotherapy, although temporary. And again, looking at the tumor from the brainstem after resection, we see what we saw in vitro, where ONCT-1 drove AKT activity here, and the combination of Paxalosib completely removed that AKT signaling. So it really highlighted the effect. And this is work done by Sabine Moore's group, who also used the combination and also showed that the combination was much more effective at extending the survival of a very aggressive DIPG model compared to the single drugs alone. And two case studies, one from Carl Koshman's group at a patient that received ONCT-1 following reirradiation and continued to experience tumor regression until she passed away of pneumonia at almost 2 years following diagnosis. And then a more recent study for a currently living patient who received ONCT-1 and Paxalosib 6 weeks following radiotherapy at diagnosis and continues to have tumor reduction almost 10 months from diagnosis. And you can see here on the last image, which was 2 weeks ago, I think, we've had a 2/3 reduction in the tumor volume from diagnosis, which is absolutely fantastic. And she's very well and back at school. So it's time to test this combination under clinical trials, and PNOC 022's opened at San Fran, and I think opens in Australia early this year. And it's a sophisticated treatment design with not only Paxalosib and ONCT-1, but some other therapies combined with radiotherapy and at different stages of diseases. But as I mentioned before, even in combination, we still have residual tumor cells living in the combination. So we need to do more. And so we know that immunotherapies have revolutionized the treatment of other forms of aggressive cancers, particularly when they're started at advanced diseases like advanced metastatic melanoma. DIPG tumors, however, have evolved mechanisms to remain invisible to the immune system. So we need to make them more visible to the immune system. And how do we do that? Can we turn off the immune masking effects of DIPG cells? And can we encourage the bone marrow hematopoietic stem cell immune system to enter the brainstem and eradicate the tumor using the natural immune response. And one way you might be able to do that is checkpoint inhibitor therapies. And I can hear you in the background that that's been tried. So what we've been able to show in work, and this is work we've done in collaboration with Pooya Faridi from Monash Health in Melbourne, is that this protein here, B2M, beta-2 macroglobulin, is a marker of response to immune checkpoint therapies in metastatic melanoma. And here you can see, here's a patient that responded to treatment with nivolumab, who has very high, and the pink is the staining here, very high level of B2M within their tumor. Whereas a patient that had no B2M, because the gene was a loss of heterozygosity of the B2M gene, meant that there was no production of the protein. And you can see here in these tumor cells, which are blue, there's no pink, which means there's no expression. And when you quantify the survival benefit given by using checkpoint inhibitor, such as nivolumab, pembrolizumab, to patients that either have some B2M or none, you can see that here, these are the patients that have a loss of heterozygosity of B2M. They survive very, very short time after diagnosis. And here, patients that have a lot of it do much better on immunotherapies. So when we analyze how much B2M there is in tumors either grown in hypoxic or normoxic conditions, so normal oxygen or in less oxygen, and we treat with ONC201, we get a dramatic increase in expression of beta-2 macroglobulin. So if we look at this visually, so these are mice engrafted with our ONC201 resistant cells, DIPG6, and we can see the blue dots here are tumor 2 weeks after engraftment, 2 weeks after successful observation of the engraftment within the brainstem, and then 6 weeks. So when we treat with ONC201, we start to see a bit more brown staining, and the brown staining is the protein, and you can see it's on the outside of the blue dots, and the blue dots is the middle of the cell, the nucleus, and the outside brown bits is where the immunotherapy target B2M would be. And then after 6 weeks, you've got this dramatic increase in the amount of B2M there is. When we treat with paxilosib, we see a somewhat similar amount of increase compared to the vehicle control, but it doesn't seem to go up too much. Again, the combination is slightly more than the two drugs alone. But then again, after 6 weeks of treatment, we get this dramatic increase in B2M expression, which means that now we're starting to prime these cells to be recognized by the immune system. So if we can combine the treatment of DIPGs with an immune therapy, then maybe we can use this as a maintenance therapy, or indeed, maybe even a debulking therapy. These are all the complex questions that we still don't know. And so if we summarize everything that I've shown today in a graphic, what does it look like? It looks complicated, I get it. But if we start at the start, ONC201 causes mitochondrial dysfunction, which causes reactive oxygen species production. And the cells respond to this in a number of ways. One, they increase the activity of this pathway, PI3 kinase signaling pathway, which is regulated or activated by insulin. And then the cells can upregulate growth and proliferation, but also antioxidant response. So if we can decrease the antioxidant response using a number of different strategies, then I'm sure what's going to happen is that we're going to get more surface expression or more expression of proteins that would encourage an immune system response. So we could then use checkpoint inhibitors, or indeed, if we're changing the surface expression of different antigens, then we could use a CAR T therapy as a maintenance therapy. And so the work that's going to underpin the focus of my group going forward is how we fit all of these different ideas into a treatment paradigm that we can get the best response from. And we've started to do this where we've challenged the cells with ONC201. We've waited a couple of days and we've assessed how much of this antioxidant response we get. And then we hit them with this drug over here and we can completely ablate them. What we haven't yet done is then subsequently measured how much of this B2M there is. And we haven't got models of DIPG in animals that we could then test CAR T therapies with. So, this work will then collaborate with clinicians that have further, and researchers that have further expertise in immunotherapy targeting, other pharmacological drugs. I've listed all the ones that I can think of at the moment here. We've got paxilosib, ONC201, valproic acid, beta-lactocome. This is a new drug from a company in Korea that we've got. I haven't tested this drug yet. And then there's all the checkpoint inhibitors, nivolumab, pembrolizumab. Do we need a CAR T therapy? So this is the thought process going forward. And I think I've gone a bit over time. So it leaves me to thank all of the people that did this work. My group, particularly Eva Jackson and Ryan Duchatel and Mika, and the lovely team, wonderful hardworking team. And then there's some fantastic collaborators, including Jason Kane, Sabine Muller, Carl Koshman, Javad, and Puya Faridi. And of course, all this work couldn't have been done without the support of families, the government. You know, the Chad Tough Defeat DIPG Foundation have funded this work that you see here today. We've got another excellent, exciting program working on Paxalicin in combination from the DIPG Collaborative. I'm funded by the government. And of course, huge foundation support from all these listed here have helped us to get to this point in less than 4 years. And I'd be honored and happy to take any questions now.
Hey, Matt, that was amazing. The big question is, at this point, if you went back to the beginning again when your daughter's first diagnosed, which treatment plan would you select for her right now? Which combination of these drugs, or radiation, drugs, PD-1 inhibitors?
Yeah, so of course, I've thought about this many times.
I thought you would, but we would like to know. I know, you know,
Yeah.
it's not official. People shouldn't be doing this, but if you had access to any drug you wanted.
Well, yeah, I think the first caveat is that we're limited by the efficacy of these drugs and the amount of it we can get into the system. And I know from parents and from trials that, you know, if you use too much Paxalicin, you get side effects and you've got to go off treatment. So Ryan Duchatel in my lab's done a huge amount of work at redoing the PK. So reassessing how much drug do we need to give it once a day, do we give it twice a day at half dose? And that work's going to come out soon, and that's funded by the DIPG Collaborative, and it's really important stuff. What all these drugs, right? We've started clinical trials without having enough data on them first. We don't know timing, dosing. So if I was to give you a nutshell, I would say I would use ONC201 twice a week, separated by a couple of days, probably Monday, Thursday, and I would use Paxalicin. I wouldn't use it on the same day. I would use it half dose, so probably about 20 mg per meter squared. I don't know if that means anything in your measurements of whatever you have in America. And I would do it twice a day, morning and night, half dose. And then once, and this isn't possible now, this is in the future, then after 2 weeks of treatment, I would come in with another drug named beta-lacocomb in hospital, infused.
Wait, can you repeat the name of that? Beta?
It's called beta-lacocomb, otherwise known as, I'll go back to the slide. Oh, it's not there. It's called ARQ 761.
Okay.
And then I would have an infusion in a hospital stay, IV, once every 4 weeks. And then I would do a scan after 6 weeks, and I would gauge how the tumor's going. And then potentially I'd even combine radiation with that, because we know that radiation requires oxygen to be effective. And from that, then maybe ONC201 and Paxalicin would work, but the side effects, because the kids will be on dexamethasone, is a consideration that we need to keep at the forefront of our mind. The corticosteroids release extra glucose, which then decreases the efficacy of Paxalicin. You know, so it really needs to be this kind of metronomic, assessed in hospital stay treatment paradigm that we have all these decision-making cues, right? Okay, patient has hypoxic tumor. So, okay, he's not going to respond as well to radiation or ONC201. Okay, we've got to start to debulk that hypoxic tumor. Maybe we get him straight onto Paxalicin. Once that happens, we start to kill off some of those hypoxic cells. We get the more respirating tumor. Then we come through with ONC201 radiotherapy, and then stick with Paxalicin so we don't enrich for a hypoxic tumor. Once we've got the debulking done, we come through and we hit them again, and then we use this beta-lacocomb in combination. And then once we've done that, and we know that there's enough immune system response from the B2M expression, we come through with nivolumab or pembrolizumab combo. Or indeed, we work with Michelle and Nick and design a CAR T that recognizes this unique surface expression profile that's caused by this increased B2M expression. So the oxidative stress that's creating this HLA expression phenotype that we've seen. We actually don't know what's being presented to the surface. That's a project in my lab by Mika Pearson, who's just joined us from Denmark after being stuck there for 2 years with COVID. And we assess what those unique molecules are being presented by the tumor. You know, they could be tumor-specific antigens, so different proteins that are produced uniquely in DIPG being presented to the tumor. We haven't been able to look specifically at what those particular proteins are, because if they are unique DIPG tumors, Michelle and Nick could make a CAR T therapy that would be super selective for DIPG cells. It would take away the side effects from GD2 CAR T. It would increase the efficacy of the B7H3. You know, using Nick's Amaya system that's locally injected, we could get rid of the barriers of the blood-brain barrier. You know, I think all of the great work that's being done around the world needs to come together in almost like a workshop where we bash out all these ideas and we say, this is what we should try. The problem is, how do you test it? How do you test it? You know, we almost need a model that has an immune system intact, that we can gauge side effects a bit better. We use animal models that don't have an immune system, although we're getting the great DIPG, because it's a human DIPG that we put in, the great DIPG response where we're seeing this immune effect. We can't now see the immune system recruitment. You know, we can't see the blood cells that would come along and try and kill the tumor because the mouse don't have an active immune system. They're, you know, immune compromised. That's why we can put human cells into them. So it's all complex. And so, you know, I'm really looking for a collaborator who has the best intact immune system DIPG model, which again is not going to be like the human disease, which is far more complicated than any mouse model that we can make. But that's where we need to go. And I'm sure this talk will help, but I'm sure collaboration with researchers around the world will help us test that in the next phase of this research. So sorry, Keith.
Well, I know that that was part of what you were trying to accomplish there at that last DIPG collaborative or the symposium there. It seems like we're kind of building a little bit of a dream team, if you will, of kind of those next steps for it. I know we have a lot of other questions, a lot of questions from the group here with it. But let me take a step back if I can, Matt. You referenced that DIPG in many ways can kind of represent almost a cancer within a cancer or a disease within a disease, and has many different elements to it. I know that some researchers look at this and they look at it more from a time and space kind of perspective and kind of see that the tumor evolves over time and responds maybe to some of the treatments there. To what effect is that going to change what we're doing in combination therapies? If we're dealing with a tumor that really isn't staying static and is changing wholly, and maybe different than any other cancer we're really fighting in that way.
Yeah,
you know,
Keith, that's a great question. And one of the things that confounds our research is that the resistant models that we have, the three of them, the three most aggressive DIPG cells from Michelle Mongi's lab, they're all autopsy samples. So they were both the two most aggressive ones that we have were both taken from 6-year-old girls who survived only 6 months after radiation. And when we look at the genomics of these tumors, they're crazy in terms of the number of different mutations. So, you know, it's an important question. And I suppose the only comment I can make from evidence that we have is that we have Josie's sample, diagnostic sample in the study, and it's quite resistant to ONC201 alone, absolutely. But it responds differently to those post-mortem, post-radiation cells quite a lot. It's a hypoxic tumor, where these ones are more like to grow in oxygen. The combination of ONC201 isn't as effective in Josie's diagnostic tumor as it is in that really aggressive model. So we don't know what the survival benefit's going to be from the combination at diagnosis. The only thing that I can say with evidence is that we've now had a patient who's had the combination. So the parents bought German ONC201, and she had mutations in PI3 kinase AKT signaling, which would suggest that potentially paxilosib could help. And she was prescribed that compassionate. And she's had 2/3 of the tumor reduced in 10 months. So it's her mutations are very similar to Josie. Josie had a few other additional mutations that this patient doesn't. But again, I agree. whether this treatment works up front, I hope so. Whether it's more going to work and provide families with a bit of extra time, maybe that's what's going to be. We don't know, and that's why we really need to do these studies on as many kids as we can at different stages of disease. And that's and Sadim Muller and the PNOC team have really designed an inclusive clinical trial that will take patients at any stage of journey, and hopefully also help us to get meaningful data from that. So yeah, I mean, it's a great question, and it's just, I suppose it's like, you know, you think about the leukemia story, where we have 90% cure rates, but we don't always have cure rates. You know, we have kids that are diagnosed with really high blast counts in their bone marrow, and they do poorly and they don't respond to standard of care. And then, you know, for those kids, which are like DIPG kids in some ways, their outcomes are still very bad. You know, I know it's only 10% or less than 10% of patients. But again, they're kind of in that DIPG boat where we're still trying to scramble. The thing is that there's been 100 years of research in leukemia to figure out the combinations. And, you know, I don't know if you've ever had a look at what a child with ALL gets, but they get chemo after chemo after chemo after chemo after chemo. And yet we give kids radiation and onc at this stage, or not even. So, you know, there's a lot of work to do, and it's just fantastic that we have such a dedicated community that are pumping money into this, and you know, the cure starts now, and the DIPG collaborative and the Defeat DIPG have put together teams of experts to give us opportunities to explore combination and other treatment ideas, and trying to get these. And without that, you know, we've done, this is 10 years of work, I reckon, at least we've done in 4 years, but thanks to collaboration and funding.
Well, and if you look at it, you know, we're seeing so many things just on a day-to-day basis. You know, you look at the survival times when we were talking 18 months as an average back in 2018. As we're getting into that 30, we're getting into even that 40-month timeframes. It's changing substantially from what we knew it was, and even what we're comparing it to. Obviously, we have a lot of specific questions. Al, do you want to go through a lot of those and try to get some of the folks' questions answered?
Well, first, I just want to go back to my original question, just 1 second.
Oh, go for it.
Is there any way to measure the response to treatment with those biomarkers using maybe like a liquid biopsy?
Yeah, so Carl Kossman's been doing a lot of this. So not the oxidative stress. That would be, so the cells would produce or pump reactive oxygen species across the plasma membrane, which is the outside of the cell. And you know what that does usually? That encourages the formation of blood vessels. So I mentioned before in my talk that the most resistant DIPGs had the most high level of reactive oxygen species, or free radicals, even in untreated cells. They were already producing heaps of it, because I know why. It's because they're trying to encourage the brain to send out blood vessels so they can get nourishment, right? They're recruiting. It's like a chemotactic. It's going, here I am, here's all this oxidative stress. Same thing happens in stroke. When you have a stroke, the cells produce huge amounts of reactive oxygen species because you've had an occlusion of the vasculature. So it's trying to encourage new blood vessels to grow around it where the blockage is. So the DIPGs are doing the same. And it seems that the most aggressive DIPGs are the ones that produce the most reactive oxygen species. Maybe there's a way that we can use MRI imaging to look at perfusion, to look at oxygen tension within the tumor. And I think that might start to give us some indications of whether the patients are indeed going to respond to ONC201 up front, or whether we need to combine it with something that might decrease the hypoxia, or indeed what we want to do. And I think this is universally right, is that what we want to do is we want to cause so much oxidative damage in the tumor that we get this breaking up and cleavage of the DNA of the tumor cell specifically. So can we harness that high level of reactive oxygen species that's already in the cell and drive it further? And that's, I suppose, in a nutshell, what these drugs and the combination therapies are trying to do. They're trying to drive increased oxidative stress, particularly and uniquely within the tumor, so that leads to the cells cutting up the DNA. And that probably also is the reason why the cells produce more immune recognizing molecules going, okay, my normal defense mechanism is, okay, I'm a cell under stress. Maybe I should be taken out before I form a cancer cell if it was a normal cell, but the cells are immune suppressed, and that's probably by the surrounding microglial cells, that there's no immune response coming in just to indeed cause the cells to be recognized by the immune system and then eaten up.
Keith, we could alternate questions. Go ahead.
Yeah, I would say we'll take questions. This question from Marcelo. He says, is there any data on oxidative burst following ONC201 and he says R cells, but I'm thinking it's R cells.
So we've measured, so we've done a lot of work on measuring how much, it's not so much of a burst. It's not like an immune system burst. So the immune system, macrophages and neutrophils do this where they cause a respiratory burst, and that's regulated by the NADPH oxidases. We don't see so much of a burst. What we see is a kind of a gradual increase in ROS. It's not a all or nothing kind of thing. It's just a gradual increase, and it happens regardless of whether the cells are resistant or sensitive to ONC201. So there's no burst, unfortunately, because I think if there was, we would be able to harness that.
Okay, next question. What is ONC206? How does it differ from ONC201?
I think it's a fluorinate. It has a fluorinated carbon, which makes it more accessible to the brainstem. And we've worked on ONC206 in animal models and in the lab, and it's about 5 times more effective. But again, we don't know in humans about the safe dose yet. That's the studies that's going on. In, I think it's PNOC 19, which is ONC206, or 23, one of the two, which is doing dose finding studies. So yeah, we don't know if it has the same effect in patients.
Matt, do you have any information on the H3F3A G334R mutation? Does ONC201 or 206 work on either one of those?
That's a great question. I think we haven't done any work on it because we don't see it in DIPG, but it's obviously a GBM. And I hate to speak out of school, but I can get back to you if Keith, you tell me who that is, because I know someone that would know.
I'm trying to see AS. I don't know, AS, if you want to go ahead and put your contact information in there, or I'll send you a message.
Yeah, great.
I'll deal with it. I'll get with you.
Thanks.
You were mentioning all this oxidative stress brings in new blood vessels. Would Avastin have a role here?
Yeah, it's a great question. Absolutely fantastic question. So Josie was on Avastin and ONC and Paxalosib for a long time. So she started ONC and Pax at 56 weeks after diagnosis, 3rd relapse, 3rd progression, and then she lived for an additional 40 months on that triple. So maybe you're right, Al. Maybe we're looking. I'm not looking Avastin. So again, a hard thing to use animal model. We have to figure it out. Someone will know. Good question. Thank you.
Keith, any more?
Sorry. Yeah, I'm trying to scroll through here. Sorry, I got caught up a little bit. Okay. Is there any data? This is kind of a different question. Is there any data on whether the COVID vaccines would be safe with Paxlovid and the ONC201 combination?
It's a great question, and I think we do see. So I don't know whether we know this directly for Paxlovid, but for other drugs like everolimus and sirolimus, the mTOR inhibitors, they have a huge effect on the function of immune cells. They're very immunosuppressive, and I worry about being on those drugs when not only when taking the vaccine, but also in this virulent life that we now live. And I send out my sincere best wishes to all the families during this time. It must be really challenging, because I know that the brainstem is really important in regulating stress response to infection. And yeah, I just hope that everyone's okay and not challenged even further by this, devastating diagnosis, but then having to battle COVID-19. So I send my message. I don't know the answer, but I can imagine that there would be some loss of immune function. We have done some data on that, some work on that using normal neutrophils and treated with the combo, and we didn't see cell death. So my guess is it would be okay, but I don't know.
Okay, for these combinations you're talking about, are they just for the brainstem tumors, or you think they'll be effective for thalamic tumors or maybe even glioblastomas?
I don't know about glioblastomas, Al. I would predict they'll work well in thalamic H3K27M DMGs. Well, when I say well, what's well? Hopefully well. And they're included in PNOC 022. I don't know about GBMs. Looking at my graph, pediatric GBM, I would suggest potentially, because they show a huge or equally high level of expression of that mitochondrial protease CLIP-P as DIPGs. So I would say there's a potential that it would work. Considering that Paxilosib's used in GBM, potentially, yeah, but again, needs to be tested under trial conditions. And we haven't done any preclinical work, I'm sorry, on GBMs.
This question is are we seeing any similarities between the resistance from tumors with ONC201 and 206? And then secondly, are there other drugs or supplements we can use to reduce the antioxidant response? One that's also mentioned is metformin. That's obviously something that's been out there and talked a lot about here recently too.
Yeah, I mean, I like that idea. I think you'd probably get more of an effect of ONC201 with metformin, and then if it was going to be rescued by an antioxidant response, you'd need to turn off the pathway that's switching it on, and that's PI3 kinase AKT, using a PI3 kinase inhibitor. But there are other drugs, you know, there's valproic acid, beta-lapicone, the ML315 drug, but none of those are accessible to patients. If it gets to clinical trial stages, then it would need to be administered in hospital. You've got to be so careful. You know, here you've got two drugs which have an acceptable toxicity profile. You know, adding anything further into there is just so dangerous. And all of these things need to be managed by the clinical team, you know, and discussed. And at this stage, we have some preclinical data, but we don't have any clinical safety data. You know, what's going to happen? Is there a drug effect, drug effect? You know, what's the timing? We don't know that. How's the white cell count going to do? What other organs are going to be affected by it? Is it going to affect cardiotoxicity? You know, there's so many questions that we have to assess under clinical conditions.
Okay, this is an interesting question. We were on ONC201 for 2.5 years, and then progression happened this year. Do you think it's advisable to get back on ONC201 and add Paxilosib, since the tumor already grew resistance to ONC201 before? Does that make sense?
Yep. If it was, yeah, well, I think you would get more of an effect by combining it with Pax than just using Pax alone, if that was the other option. I mean, there could be other much more useful options at that advanced stage, but
Like
because I don't know. Yeah, there's 1,000 drugs. Right? I get an email every week listing another clinical trial.
Yeah.
You know, that's up to your clinical team. I can't speculate, and I don't like to shoot down anybody else's work. But if you were going to go down that path, then I would suggest that probably the combination would be more effective than the monotherapy.
How about adding re-radiation too?
Yep.
You wouldn't try the ARQ 761?
You just can't.
You can't get it.
Yeah, you can't. You can't. Okay. I mean, yeah. In time, hopefully, that's where we need to go. And I've got to spend January writing some grants to try and, you know, get this next. So this is the end of the Michael Moisier Defeat DIPG fellowship that I've been on, and now I need to find another way to keep this work going. So that's what I'm doing all January, writing grants to do the ARQ 761 and the valproic acid and the checkpoint inhibition. Yeah. Okay. Because the Australian government just doesn't fund it.
You brought up re-radiation in this, and obviously it's something that we talked a lot about here recently, but do you see that there is a role of re-radiation in some of these combinatorial treatments? And does it effectively present maybe a pause or a reset button in between some of these treatments, or do you see it as just kind of augmenting what's already there?
I think it has an important role to play. I think the timing of it is important too. Yeah, I just think that maybe we wait too long. and we need to do the appropriate clinical trials. But it's pretty guided now. Stereotactic radiotherapy. Unfortunately, it is challenging, a challenging experience for the child, and I recognise that and know it firsthand. I was there with Josie both times, and I don't in a situation of such challenging helplessness. I hate to subject our kids to any further discomfort. I get that, but I kind of wonder whether we're waiting too long. So for some of the responders that we get from these single therapies or combinations of therapies, whether we're waiting too long. Like if we get a response and we get tumor regression and we're out to 12 months, should we not be hitting it again? I don't know. This kind of gets to me a bit. We know that it's the only recognized treatment. So why do we wait? And these are discussions to be had with clinical teams and things. And I know it's not the protocol, but again, let's not forget that the protocol doesn't work. We don't have any survivors. So it's not a protocol. It's an experiment within the healthcare system that's funded by the healthcare system. It doesn't work. So let's rewrite the protocol, but get some data from it. And I suppose, a clinical trial needs to be had using reirradiation before progression. That should be, that's a clinical trial that should absolutely be done, particularly for responders, and to see whether we can get some cures. Well, when I say cures, 2 years, 4 years, these things that none of us had the luck, didn't get to enjoy. Because who knows what happens in 2 or 4 years? Nick and Michelle's CAR-Ts and these combination treatment stuff. We've done 10 years of work in 4 years. What happens if you can give your kid another 2 years? Where are we going to be? So I totally agree. It's a conversation you need to have with your clinician. But I think it definitely has an important part to play in the treatment going forward.
Okay, you covered this before, but there's confusion in the group. Is the German UNCTOR one the same as American? And has it always been that way?
No, so it wasn't. It was slightly different until we got a capsule. So this work was submitted mid-year, and we got a capsule in March from a lovely family in Japan, and it looked different. And he'd sent us the original, which looked the same, and it was the same when we tested it, and then got another batch in March this year, and it looked different. So we did testing on it, and it was the same as it's now the same as the American. So it's now the acid as opposed to the base. So once we saw that, we didn't do any more testing in terms of efficacy, because that would be repeating that whole study again. And we've got other things to do. Where do we go now? Is that where we need to go? And I'm pretty sure, and we've only tested one capsule too, I must say, but it was the same, exactly the same as the Chimerix UNCTOR one or Oncoceutics as of March. So if you've got capsules post-March, then it should be.
Any thoughts to other types of combination things to add to the combinational treatment? I know there's obviously data on ACT001, AMXT1501, CBL0137. Has any of those been discussed as possible combinations with obviously the more well-tolerated ONC201 at this point?
No, and that's the thing we need to do, Keith. We need to almost have like a focused summit where we don't give presentations. We just sit around a round table.
I tried to get you to the States for that.
I'd love to sit down with David Ziegler and his fantastic combination strategies and see where some of these fit. I suppose David's leading the way with Michelle in terms of the combination strategies. I'd love to see how we can make them work even better. But with David's AMTX DMFO, the in vivo animal study data is the best we've seen of any combination strategy. So I suppose now we have to see what happens under clinical trial conditions, and which is what the Connect Network and Cure Starts Now and Collaborative are supporting. So yeah, I mean, let's hope that does what it does in animals, does it in humans, and we now have a treatment. But if not, we need to do this cross-contamination and have a look. So, I haven't assessed the response of those cells to those drugs the same way that we do this. And this is this pharmacophosphoproteogenomics stuff, Keith, which is a mouthful, but we really assess the genomic and the proteomic influences and responses to drug treatments. And that's because, my forte in research expertise is in protein sequencing. And so I'd be happy to do those techniques on any of these combinations to see whether these additional arms or these additional drugs that we know can get into the brainstem may help. And then it comes down to timing. What's first? Do you do it together? You know, there's huge amounts of studies to be, each one of those studies are huge. Anyway, yeah, love to combine them and see what happens. But I can't give you an opinion on it because I don't have any data.
I'm just about out of questions, Keith.
Yeah, I mean, they're kind of running together at some point here. And a lot of them are dealing with, okay, what about this? What about that? And there's going to be a lot of this I'm not sure we're going to have all the answers to at this point.
No, and I won't have the answers either, yeah.
Yeah, and so, I mean, in looking at this, I think the model that you have and really what you're doing and really starting, I guess, with ONC201 and various combinations therein, because it's kind of our baseline here at this point. But then taking it to the next steps would be really helpful. I do want to ask you, though, a little bit about to what degree, and this was a question that was posed in the thread here as well, to what degree some of the model and some of the ways you're doing it is similar to what, you know, a GBM agile is doing. And is there things that we can learn from those that we can apply to these combinational frameworks, if you will, not just trials, but effectively how we deal with all of them?
Yeah, I mean, at least there's a trial design now that's kind of adaptive in GBM agile. But again, I think that's somewhat also recapitulated in DMG Act or the new PNOC 022, which is an open forum for additional drugs to be added and combined with, you know, we're using ONC201 as our backbone because of the safety toxicities. And, you know, you've seen all this data. And Carl Koshman's got a heap of data as well about EGFR in terms of how it drives response, you know, to ONC201. There's kind of a body of work that's been done now by a number of labs that kind of gives us the impetus to push forward with the one starting point. And then how do we go from there? You know, we need a starting point, and the starting point's always radiotherapy. And then there's what happens next. And what we add to that is what we're doing. And I suppose in some ways, PNOC 022 is like GBM Agile. It's just what we need to do, and when we're trying to do this with our DMG Act collaboration, is to bring other combination therapies into that, be tested in other labs than the labs that it was discovered in. That's what happens with all this data. All the in vivo stuff's tested in another lab, just to make sure that it works across models and it works across labs and stuff, which is a really important part, I think. But I think, you know, we kind of have that in some ways, but we just don't have the drugs. And so all the drugs that are on GBM Agile, one of them is Paxalicib, right? So that's the drug that underpins it. And we know that Paxalicib, at least in our models, works better in DIPG than it does in GBM. Whether GBM Agile needs ONC201 in it, or maybe it does with Paxalicib, but of course, you know, we're not necessarily linked to that study. And of course, I'm working with some GBM adult clinicians who are seeing more and more young adults being diagnosed with DMGs. And I've seen quite a lot of it lately, particularly here in Australia, which is really sad. But they don't have the options at all. You know, often they're not biopsied. They get temozolomide every time, which doesn't do much. And then, you know, they're left with pretty much nothing. They might do a resection if possible, but again, in the thalamus, it's very challenging. And so what we're trying to do is, I mean, DMG Act or PNOC 022 is open to adults up to 39 years of age, but the problem is that we've opened the trial under the pediatric centers. So now we have to work with our adult colleagues to get it open for patients with DMG, and in Australia at least, and we have to work with the pharmaceutical companies to see whether that's possible yet.
Okay, well, we appreciate you joining us here and presenting on this. We're running out of time here at this point, but obviously this is going to continue. It's going to continue with a lot of research, and you know, thank you, Dr. Dunn, for being part of this. And we appreciate obviously the partnership. This is a partnership that's even bigger than all of us, whether it's Run DIPG, whether it's Misella Foundation, who brought this learning session and webinar to the front, or even the DIPG Collaborative. We all work as one big group. So the discussion still continues, and we ask that if you have any interest in learning more about it, these webinars are done. How often, Al? I know you post a lot of this on your website.
Yeah,
But,
whenever we need one, whenever we have a good topic. We also do them every week during Brain Tumor Awareness Week. But this was an important one, so I said, as soon as we could get it going, do it.
Yep, and encourage them, follow them. This is how we learn. So, well, thank you very much, Dr. Dunn, and we appreciate your time here tonight.
Thanks,
Thank you all.
everybody.
And may I send my sincere best wishes to all the families that are fighting the journey, and to those that have lost their most important loved ones. My heart's broken with yours, but we will continue fighting, and thanks for the support.